This gazelle is partaking in its everyday meal, but little does it know that today it is being watched by a fearsome predator. At any moment, this gazelle has to be ready to push its body to the limit to escape being a cheetah's dinner. As the cheetah explodes into action, the gazelle... That jolt which you have just experienced was also felt by the gazelle as its brain recognized a dangerous situation. When the brain senses and recognizes an intense situation, it sends a signal to the adrenal glands, which are located on the kidneys. The hormone epinephrine is then produced in the medulla of these glands and is released into the bloodstream. Epinephrine will then act as a ligand, a signaling molecule, to trigger multiple responses within our body. Since it travels throughout our whole body, it is considered long-distance signaling, which is called endocrine signaling. The epinephrine hormone is polar in nature, so it cannot diffuse through a cell's lipid bilayer. It communicates its presence by binding to a specialized receptor protein which is embedded in the phospholipid bilayer. In a liver cell, the epinephrine ligand will bind to a G protein coupled receptor, which will cause the receptor to change shape. A G protein, which is bound to a GDP molecule, will then be able to bind to the activated G protein coupled receptor. Once the G protein is bound, its GDP molecule is replaced with a GTP, and so the G protein is now activated. This newly activated G protein will travel along the cellular membrane till it reaches an adenylyl cyclase. Since the G protein is now activated, it is able to bind to the protein adenylyl cyclase, which causes the adenylyl cyclase to change its form. In its new form, the adenylyl cyclase is able to facilitate the dephosphorylation of an ATP molecule, creating a diphosphate and a cyclic AMP, which is also known as cAMP or CAMP. The CAMP molecule then proceeds to diffuse through the cytosol until it finds and binds to a protein kinase A. The binding of a CAMP to a protein kinase A activates it. The activated kinase A then proceeds to dephosphorylate an ATP and donate a phosphate to a phosphorylase kinase, which changes its form and activates it. The phosphorylase kinase is then able to bind another ATP molecule and the protein glycogen phosphorylase. Once the phosphorylase kinase phosphorylates the glycogen phosphorylase changes shape. The glycogen phosphorylase then begins to break down glycogen into individual glucose molecules, which are then released into the bloodstream to be used by other cells in the production of ATP. The signal, however, does need to be terminated or it can result in some serious consequences. There are two ways that the epinephrine signal in a liver cell can be terminated. First, the epinephrine ligand leaves the G protein coupled receptor, which causes the receptor to return to its former shape. The second way is that the adenylyl cyclase acts as a dephosphorylation agent and cleaves a phosphate from the activated G protein's GTP, which causes the G protein to become inactive and unbind from the adenylyl cyclase, causing the adenylyl cyclase to return to its former shape.